vitally important that companies like ours across the United States receive the kind of funding and investor interest to bring forth initiatives and breakthrough technologies that can change the lives of Americans in the future.
Well, I think it's quite common that we get an interesting biological molecule from nature, but that's not optimized for human consumption. It's really optimized for the benefit of the plant, for example. In the case of cannabis, it's really to protect the plant from oxidative stress or from getting eaten by insects, and not really to be consumed by a human and be distributed through the body.
So we wanted to make CBD a more water-soluble compound to improve bioavailability, and that's really important because it does have good pharmacology, but we wanted to improve upon that.
So why am I excited about this compound? It's not about wishful thinking or a matter of faith. It's a matter of seeing the data.
Yeah, and what really surprised us about KLS 13019 is not only does it mimic CBD in that it can prevent neuronal damage, but also it's profoundly anti-inflammatory and quite a bit more than CBD. And then beyond that, we think it can be anti-addictive. We have indications that it can decrease craving for opiates. So if you have a pain medication that also treats the inflammation, let alone addiction, that could be a really powerful therapeutic agent.
We've been fortunate to work with truly insightful and innovative and experienced medicinal chemists who knew the literature well enough, know what not to touch about our initial, but found an area that actually could stand improvement. In this case, we just wanted to make the molecule more water soluble. And what we got was vastly better than that. In addition, we went from looking at something that may have 50 targets to has 2. So it has dramatic pharmacological specificity. And frankly, it's just a better drug.
We started to compare our compound to CBD, and that's where Temple University had some pre-existing knowledge of the work that we were doing and enabled us to get into a phase 1 study grant with them in 2017. So we quickly found ourselves going from neuroprotection with our lead compound into a potential source for non-opioid based pain management.
It was critical to have the resources to do actually the more expensive aspect of drug development, and that is studies of safety. And that box now has been checked through the granting agency. Having said that, we're still are looking for opportunities to look at other related areas into neuropathic pain to broaden our scope and net in looking at other therapeutic areas.
The phase 2 STTR grant was really a profound impact on our company. We were really relying on it to advance our compound into phase 1 clinical trials.
Our compound interacts with none of the opiate receptors. We have demonstrated that, and the NIH has demonstrated that for us.
The whole point of the HEAL initiative, which is help end addiction long term, or helping to end addiction long term, was designed so that the removal of opiates and the market use of anything other than palliative or end-of-life care can be substituted by new technology.
Pain is, particularly neuropathic pain, is a huge unmet medical need, and be it from any number of sources. The one we're focused on, of course, is chemotherapy, but it's certainly not restricted to that. We have the potential here of helping millions of people dealing with pain and potentially addiction. So I've been in this business for 45 years, looking for ways which to facilitate the brain and helping itself from various kinds of pathological problems. After 45 years, I have not found anything that does better than this molecule.